It's a pleasure to introduce Sandy Anderson. We all know and love here in the Math On community. Sandy and I, as you might have heard, had known each other for 20. Did we decide it was 23? Yeah. 23 years. Okay, 2000. Okay, so 23 years ago. But the first time I ever spent any significant amount of time was I visited him in Dundee. And my favorite story has to do with them asked what to do that night. And he was off doing something. He suggested going to this play thing. Okay. And I was with a much older colleague who's a chair of neuropathology. He was roughly 80 years old. He and his lovely wife were there. It was delightful. So we go to this event, and I can't really explain the venue because there are just no words to explain this place. And it was supposed to be some sort of comedy, something, but it was like mumbling, impossible to understand Scottish slang, which I could hear all the cursing on repeat. So it was like infinity cursing. And it turns out it was train spotting. Yeah. Yeah. I don't know. All I know is the 80 some odd year olds left the room, and I was just, it was one of the many fascinating experiences with Sandy.
Thank you. Anyway, that was a good play. There was a lot of swearing in it though. And if you've seen the film, it would seem appropriate, right? Anyway, so I know that I'm talking about agent-based models, but I can't do that just as a generic thing. I have to talk it through a framework. And to me, the most obvious place to start is really thinking about the cancer ecosystem. And this is something that we've been, harping on about probably for about 2 decades. And in part, it's because of David Basanta. It's largely his fault, because when he came, and he's like, we need to include all these other cell types and think about how they interact. And oh, by the way, it all has to be homeostatic. And I'm like, what? That's going to be impossible. Because he was coming from computer science where they were evolving these tissues and making them homeostatic. But the more we worked on it, the more we realized actually homeostasis is a really important constraint on any cancer system, because the cancer is invading that normal organ. Right? So tumors don't grow in empty space or in serum conditions. I'm sure most of you all know that, but we might use data from some of that, and we might even simulate that. Cancer isn't only a collection of mutated cells. I think we can agree on that. Cancer is a complex evolving system of many interacting cellular and microenvironmental elements that were once normal. And often that transformation is still surrounded by a whole bunch of interacting cells that are still normal. You can literally have 40% of your lung filled with cancer and still able to breathe. It still functions, right? And so that's one of the crazy things about this disease that we're all working on. And this is a beautiful cartoon from one of David's postdocs, who was particularly artistic, Arturo, talking about the bone cancer ecosystem, just as an analogy for what we're talking about. So a little bit of, in some sense, philosophy, what have we learned over the last 20 years thinking about modeling cancer and maybe thinking about the ecology of cancer? And I would say that something that's really come forward for me is this idea of using models to either generate hypotheses or test them. And they can do both, but often you start from one of those points. And so often you might get an unusual result in an experiment, or you might have some really cool data, and you're trying to understand why it's doing what it's doing. And so often in that case, the mechanisms might be unclear, right? But the way that you're going to make sure that the mechanism you think is at play is you're going to calibrate it precisely to that data and say, oh, it's a good fit. So that could be one of the methods that's driving this behavior, or one of the mechanisms. On the other hand, this more hypothesis generation approach is kind of like, we have an idea of the underlying mechanisms, but we don't fully understand how they might connect. We might not have a lot of good data, but we can have ballparks. We know it needs to be homeostatic or whatever. So we'll often play with these, what we call phenomenological models. They're oversimplified. They don't capture all the elements, but they actually give you cool insights. And sometimes those cool insights then drive experiments that then can feed back on refining that model, and there's a loop. So it's just worth thinking about that. I really consider cancer as an emergent phenomena. It isn't, it's not it's sufficient to just have a mutation. And I mean, it depends how many mutations that the cell might have, or a bunch of cells might have. But we know now that normal skin and normal tissues are actually littered with mutated cells, and they're not turning into cancer. We've all got moles that have all the mutations that are required to drive cancer, but they're not turning into cancer. So there needs to be a triggering event or an additional event of some sort. And one of the kind of aspects I think that's really important there is this idea of homeostatic regulation, of control of the normal system. And so our tissues and all our organs are really robust to being wounded, cut in half, taking one out, putting one out, taking someone else's organ and sticking that in. It's amazing how robust we are at that. It's incredible. And one of the things that's become clear as we went down that path is, how do we actually measure that in an experiment? How do we quantify that? And in many ways, it's an impossible experiment. You just couldn't do it. So that's where I think modeling particularly comes in as a really important player. And this whole concept of really understanding processes in isolation is sort of, the reductionist paradigm in biology, but it's also a necessity, because you can't measure 10 things simultaneously, because you'd have to destroy the animal and then regenerate it from the exact same time point magically and then remeasure, which is an impossibility. But models can reconstruct the system. They can piece together those individual elements and integrate them into a system. And you then can play that forward and say, well, each of the component parts are doing what we think they should do, but when they're together, they do this weird thing that we've never seen in the animal or whatever. So there's something not right about how we've wired up, and you can play with that. You can change that. You can tweak that. And one of the tweaks is that system should be homeostatic in the absence of that cancer. So the way it's connected, the way the components interact and the processes that drive them, if they maintain kind of a regular turnover that isn't exploding in growth or disappearing, that actually helps constrain the system. So I'm going to talk about a couple of fairly complex models, but homeostasis is going to be one of the underlying features that kind of keep them together. And of course, dynamics, which is really about almost all the models that we've been talking about, that can really also be insightful. How those things change over time and space is really critical. And again, that's something that's really quite hard to do experimentally, although with all the new technology, we're starting to make strides, right? So I did want to point out that we did build some software and some methods, and Rafael Bravo, who's in the room, where is he, Rafael? Put your hand up. That's the man that wrote this in large part. And you can go there, Hello World, it's HAL. And of course, it's a little play on the sci-fi computer in 2001 that doesn't do what you tell it to do, right? In fact, it tries to kill you. That's our CA system. And it's on and off lattice models. And there's lots of other cool tools out there like Physicell and CompuCell and so on. The purpose of HAL was really to be able to very quickly generate simple cellular automata models easily with a visual interface, right? It just does it quick. You can add more sophisticated physics and so on if you need to, but that's not really its job. So everything that I'll talk about today wasn't generated in HAL, but it could have been. The reason it wasn't is because we didn't have that framework yet, but they're all using a similar sort of framework. And so the first little vignette I want to talk about is in regards to prostate cancer evolution. And we've already heard a bit about the role of stroma, and in particular, I'm going to focus on what's called reactive stroma. And so you're probably wondering what that might be. So in prostate cancer, there's this grading system called the Gleason score, and it was defined in 1966. And that literally, that graphic there is part of that system. That's how they identify what their score is. And they take two different samples and they say, well, it's kind of a bit like a 3 plus a 2 or a 4 plus a 5. And that gives you your Gleason score. And it's a completely tumor-centric test, like grading. It ignores, it doesn't include anything about the context. It's irrelevant as far as that system's concerned. We think it needs to be updated, but it's still something that we use. And if you've got bad Gleason scores like 9s and 10s, then that's bad news. And if you've got low Gleason scores like 4s and 5s, that's good. Anyway, so the point is that we need to improve that. And working with a pathologist, Gustavo Ayala, this is some of his images. This is like biopsies from stroma that's beside tumors in the prostate. And he's sort of coloring it according to the abundance of reactive stroma. So the amount of blue in this image in each one of these cores highlights how much reactive stroma there is. And so you can see in that top panel there, 0 and 1, there's less than there is in the bottom 2. Right. So the point is, in patients, there's a diversity of reactive stroma or stroma that's surrounding the tumor. So, you know, we wanted to try and understand that relationship, and we dug in a lot to the sort of biology around it and came up with this interaction network. And recently, Jeff West, who's sitting back there, and myself and Mark Tessie wrote a little kind of perspective piece for Trends in Cell Biology, where we talk about how agent-based models can facilitate integrative science. And this sort of idea of a visual interacting network of the different component parts is like the perfect tool to really drive that dialogue and that process. So basically distilling down what are the key elements that we need to worry about, what can we actually measure, and how do we think they interact? And so a green interaction is positive, a red one is negative or inhibitory, and then we have these rainbow interactions, and those are actually the fact that the tumor is heterogeneous, and so it can produce a whole spectrum of those factors. And so, you know, the different cell types there, many of them are normal, and many of them are also going to be transformed or modulated by those other components. And so what we do is we take that network, and then for each of the cell types, we build essentially a flowchart life cycle. It's like a decision tree for what that cell does who it interacts with in its environment. And those decisions then make it divide or die or mutate or consume or produce some factor. And I don't want to go into all the details of that, but you can see it's fairly complicated. But a lot of this was driven by trying to understand the biology and working closely with our biological collaborator, Simon Hayward, and our pathologist collaborator. And we argued a lot about it. I'll be honest, like almost physical fights about some of the components. Never came to blows, but it was an intense discussion because even the pathologist and the biologist couldn't agree on what was important and for some of these things. And so, but the point is that once you actually articulate a network like that, it really clearly brings together a perspective from that group, and it helps you actually see what you might be missing, what you might need to add. Because it's hybrid, we also have some equations here representing the continuous variables, and these are obviously going to be oversimplified because we're assuming everything just diffuses and is produced by the appropriate cell types and so on. Like, you know, the ECM here is essentially just a non-diffusible field. And we know that there's mechanics and other complicated things that could be integrated, but I'm not doing that. So anyway, we take that system, and what we had was, because working with our pathologist, is like all of these cancers occur in what's called the peripheral zone. And if you look at that small pink thing, that's actually a prostate, and that small box in there is the peripheral zone. And so we take that and we kind of digitized it above there, and it turns out to just be under a square centimeter. And then we kind of put all our cell types. We've got basal cells, luminal cells, immune stroma, and essentially created a 2D virtual tissue, right? It's 2D. We know in reality it's actually 3D. So that's already an oversimplification that's quite major, but I'm going to do it. And so we take that, and then what I can do now is initiate a tumor in there, and that tumor will start producing growth factors, TGF-beta, for example, that will then excite or react the stroma around it and cause a reactive stroma interaction that then might also trigger others. And so I just show you, like, if we just look at the reactive stroma versus the tumor, and then you can see what's an interesting prediction right away from this model is that the reactive stroma can drive reactive stroma. So you've got yellow areas that are nowhere near the tumor, but are being triggered by the stroma themselves. We never actually proved that was true, but we think it's a kind of intriguing prediction, right? Worth thinking about. So what we were really kind of intrigued about was if patients have different Gleason scores, can they have different reactive stromal grades? And that's what my pathologist collaborator discussed. And is there combinations which are very bad and combinations which are good? And could that help us decide whether a Gleason 7, which is actually quite a difficult scored patient, to decide whether we should remove it or we should just leave watch and wait, which is the classic way to treat prostate cancer. So that was one of the things that we're interested in. You're going to see these sorts of images, reactive stroma, growth factor levels, and tumor cell phenotypes, and I'll explain what those are in a second. So this is in what's called a low reactive stromal environment. If we just focus for a second on that pink box there where it's reactive stroma versus tumor. And so yellow is reactive stroma and the tumor is this dark brownish color. And then we look to the far right-hand side and there's the tumor cell phenotypes. And the tumor cell phenotypes here are whether they're producing more growth factors or more proteases, more matrix degrading enzymes, whether they're dividing slower or faster. And so I'm coloring it in a very crude way, which is, let's call it aggressiveness, right? There's all the details are in the paper. You can look exactly what it is. But you see that when I rerun this again, what happens is that they're all blue to begin with, those phenotypes, and over time, the average phenotype drifts up to being a little bit green. And if you look at that bunch of colored cells there, there's a bit of green, more green than blue in there, right? It's kind of shifted. And then up there shows you the growth factors. So this is a low reactive stromal grade tumor, and it was a 7-year simulation. And now let's see what happens if we have a high reactive stromal grade tumor. First of all, you see way more yellow occurring there. And then on the right-hand side, what was surprising was that the tumor cell phenotypes didn't quite shift just as much, right? You saw that there's less green there, right? So that seemed a bit counterintuitive, and it also created a huge argument with our colleagues because they're saying that's not right. The more aggressive must have higher, must evolve further. And I'm like, well, you're saying, we're not saying it's more aggressive. They're starting identically here. That's one of the beautiful things about modeling. All I'm changing is the reactive stroma. That's my only change. And so we can actually look at that now if I let them both grow to the same size. And we do that where we initiate that kind of tumor phenotype in terms of different levels of aggressiveness. So this was actually the thing that satiated that debate, which was, okay, we'll show you, we can start with every one of those tumor cell phenotypes as our initiating tumor, and then we'll vary whether the amount of stroma is high or low. So active RSG3 is high reactive stroma and RSG1 is low. And one way to think about this, and if my pathologist colleague was here, he'd kill me for saying it, but the way I like to think about it is imagine below there is like Gleason grade, and blue is low Gleason and red is high. Then it's kind of telling us that for the phenotypes, not for the RSG, as I increase my Gleason score, I'm having less impact from the reactive stroma. There's some middle Gleason score where there's a huge difference between reactive stromal grade 1 and reactive stromal grade 3, right? You can see the space there is kind of quite different and the colors are quite different. Okay, so we can also look at that and see, well, what other stuff's going on. And here we can see, we measure invasiveness and we can also measure their survival. So what's interesting is that those RSG3s are way more invasive. So higher reactive stroma is much more invasive than low reactive stroma tumors. But it looks like the RSG1 survives better than the RSG3. So there's all this kind of slightly confusing, like, phenomena. So we turn to the data. One of the great things about working with Gustavo was he was a pathologist, but he'd worked with the same surgeon for like 30 years. And so like they had this, the exact same surgeon had removed like 900 prostates, and we had like 20 years of follow-up on what happened to those patients, but he also had the whole prostate. So we took those prostates and we segmented, like, in that peripheral zone, and then we colored them according to whether it was tumor, normal stroma, or reactive stroma, right? And so that's what you can see in this picture here. And so then if we quantify what happens when we look at that in terms of the standard Gleason categories, so right now I'm ignoring stroma, I'm just saying, tell me what the Gleason score is, then for those patients, it's very clear that this is a Kaplan-Meier curve for you, those who have not seen it. Each drop is effectively a patient dying. And the red curve here is high Gleason scores, so between 9 and 10, you can see that. They die within 200 days, but all of them. But if you see Gleason score, the moderate, the 7s are a bit longer, and then the 6s, most of them are still alive after 250 months. So what happens if we split each of those into high and low reactive stroma in terms of their abundance? And what's kind of cool is that it segregates every one of those Gleason scores. And in particular, one of the tricky things to segregate is Gleason 7s. As I said, some clinicians will say we should remove a Gleason 7 tumor, and some will say we shouldn't. And so if this could actually help you decide whether you should or shouldn't, and in particular, look at the dark green line there and the light green line, and you can see there's no overlap between those two populations, right? They're quite distinct. So this idea that combining a kind of phenomenological interpretation of what reactive stroma is with a real quantification of what it is in tissue, and using that to segregate some patients and then maybe make decisions whether they should be treated a certain way or not, was kind of pretty cool. And one of the even cooler things was, because we had these biopsies, we could actually look at the individual cells, and we stained them for the kind of 3 major, I guess, players that are involved in prostate cancer. And these were AKT, androgen receptor, and NF-kappa B expression. And so when we look across, so up top here is RSG1, so low reactive stroma, RSG3 is high. And if you just look crudely, like red means there's a very high level of that. There's much less red in RSG3, right? So now, things are starting to come together. I'm thinking, oh, there must be a reason. Why would you express less of the bad stuff if you're surrounded by all of this reactive stroma? And so what we realized was, or what we kind of concluded from this, is that if you look across all the patients, and each one of these lines is a patient, and of course we had multiple biopsies from that individual core. Blue is where the Gleason 7 would be with low reactive stroma, and red was where it would be with high reactive stroma. And you can see the red is below the blue in terms of the expression for across all those patients, right? If you look at Gleason 8 and 10s, though, there's no statistical difference between red and blue. They're just completely overlapping. And then there's some difference in the 6s. So what's kind of, for me, the implication there is that in those intermediate Gleasons, the reactive stroma is actually having some sort of suppressive effect on how much of these proteins that those cells are expressing. And so why would that be? And so our kind of conclusion here is that the stroma is actually providing an ecological niche, a support for the tumor, so the tumor doesn't need to evolve quite as far. It's like, I'm in this really lush environment, I'm surrounded by that, as opposed to I'm in a really tough, harsh environment, I'm not getting those growth factors, I'm not getting all the goodies, now I have to evolve. And that's what we're saying is the difference in these two things. And so we've did subsequent follow-up work, and there's some experimental validation of this in animal models. But the kind of concluding visual here is that reactive stroma is definitely a regulator of prostate cancer progression, and it might also impact other cancers. So that's one specific example of how we've used basically CA models on, in many ways, a very different scale to where the data was at, and made some conclusions that then allowed us to look at data through a different lens. So in many ways, that was a kind of phenomenological sort of approach, right? So in the second vignette, I'm going to talk about dissecting the impact of signaling heterogeneity on targeted therapies. And this is a non-small cell lung cancer biopsy, and it's been stained for EGFR expression. And if you look at it, there's a bunch of green stuff, and there's some little red areas too. And so the red is where it's actually particularly high. And in those reddish areas, you can see one in the kind of left-hand corner there, there's actually a bunch of stroma. And some other bits, there's stroma there's this big island in the middle. Do you see it? That's stroma. And so one of the kind of interesting things that caused us to look at this in a different way was there was a bunch of papers had come, well, before I get a bunch of papers had come out that said that, you know, secretion of HGF could influence the way those targeted therapies work. So if I happen to be beside the stroma, and of course, Andre's already articulated beautifully the potentially protective role of stroma. So I thought, could we do something with agent-based modeling there? So we've all heard about the genotype-phenotype relationship. And a few years ago, when I was a bit younger, we were determined to tackle that question. Of course, it's the impossible question. But we used sort of neural networks as a means to map functional input from the environment to functional output, which would be phenotype, and then just sort of created this blended, mixed bunch of hidden layers that represented the pathways. And it produced some interesting results, and I'm not going to talk about that today, but here what we really wanted to do was actually have a real pathway between the environment and the phenotype. And so this is kind of the way that I think about that relationship between genotype and phenotype. And it's a bit simple, right? And a bit abstracted, but that's how we did it. So, oh yeah, and this whole kind of environment mediated resistance and the role of potentially, you know, matrix driven secretions that might come from, well, from fibroblasts or from other cells that are present. And so if you actually try and look at where for this case, it's non-small cell lung cancer, and then this is like I think this is the EGFR or like one of those signaling, the RAS pathway or something. And it's just that's me only cutting it down to like 100 nodes or something, right? In reality, it's tens of thousands. I'm like, well, we can't do that. I mean, I can't possibly model that. That'll be useless. So I'm like, what can we actually knock down in an experiment with these, right? And so when we did that, then all of a sudden the network became a lot simpler. We threw out all of the stuff we couldn't actually measure or control, and we distilled it down to this, what we call minimal network. This is a recurrent network, so it has a loop if you look closely, so it could keep going on forever. So we'd have to wait for it to come to a steady state. But essentially, green here is a kind of, you know, promoting interaction, and red is an inhibitory one. You see there's only one red on there. The greens are varying thickness. So one of my experimental colleagues at Moffitt, Eric Hora, had the husband of my postdoc, Unjun Kim, working in his lab. And we managed to convince Eric through basically the husband to try and measure these things. So let's take every node in that network and inhibit it with a drug, and then look at you know, the expression of what those other proteins do when you do that. And of course, this is a few years ago. This paper is quite old. So we're using gels. And now with this network, could we actually, with this data, could we now go and train that, calibrate that network? And that's what we did. So, you know, we took that neural network and we used some optimization approaches. And then we were able to essentially calculate all the weights such that they would reproduce that data. When I knocked down one of those nodes, it would show me that network. Now, you can see that some of those have a few error bars that are a little bit bigger, and it's not all perfect, but it's pretty damn good. But what we decided to use was not like, there isn't any single best network. There's probably about 500 that are really, really good. And then there's thousands that were just crap. So we took those 500 and we kind of turned them into like virtual cell lines or virtual little signaling networks, right? And we say all of these describe the data well. So let's do some stuff. Let's treat them as a population of cells and inhibit with those drugs. And it kind of does what it should do, right? But you can see it does so as a distributional response. It doesn't do exactly that way, the bar where it should be. And the line on there is where it should be, right? And as opposed to where it might be, where it actually is. And so it was interesting that the ERK, it was way underrepresented, the MEC, it was way underrepresented, but the bulk of the rest, it wasn't doing too badly. Anyway, so we took that and now we thought, okay, well, we've got this network, let's do some combination therapies, right? Why not? And so that's what we did. And so what you can see here is the more negative that is, the more likely it's that population is going to die. And so you can see the kind of monotherapy in blue, then we add ERK, or then we add MEC, and we do that all the way throughout. But the real kind of intriguing part was then, okay, it gave us some specific suggestions about which combinations we should do. So we then went and did that in the lab, and you can see the comparison from the experiment to what the prediction was. And we didn't do too badly. We did over predict for some of those, but it was surprisingly good. And this, remember, was trained on knockdown data for single parts of that inhibitor. We weren't given it combinations. So you're probably wondering what that's got to do to context, but we thought, well, there must be something in this signaling. Now let's try and analyze it. So we dug a bit deeper into all 500 of those virtual cells and did some clustering and tried to see, well, is there something special about what those, how those cells respond? And clearly there is a whole bunch of clusters there, and there's definitely every possible combination. That's what that is, all the possible drug combinations on the left, and that's all of our virtual cell lines clustered to the right. And then you can see whether you're dead would be blue, and if you're very much alive, you're red, and if you're yellow, some of the times you died and some of the times. And so you can see there's definitely a whole bunch there that are really good at killing you. There's also a whole bunch that would just be a waste of time. And in fact, some would actually make you fitter. That was the implication right now. We didn't test that, but I thought it was kind of intriguing. And then, Unjung was really good at creating sort of these circles plots, and we thought it'd be cool to see how the weights of the network were distinct if we looked at either side of these. Now, remember, this is an in silico signaling network that's oversimplified and, a cartoon of reality. But because we have 500 virtual cells, we can actually look at quite distinct responses there and say, what's different about those networks? And could that be insightful? Could it be useful? And so you can see the weights are very different between this cell line, cell A and cell B. And in particular, there seems to be a much stronger influence of the RAS wild type in A compared to what it's doing in B. Anyway, we could go on and look at these things, but to me, the kind of intriguing implication was when you start messing with now their context, in this case, HGF, which we were imagining would be secreted by the fibroblasts, because that was the thing that maybe I missed. I didn't tell you completely when I showed you the data. We had the knockdown with and without HGF, right? When we inhibited those with those drugs. And so now if we look at cell A and B, two distinct, you can see that the HGF has had a much bigger impact, and particular on MET compared to the RAS wild type. So it's really quite a different network that emerges from under HGF to not under HGF, right? And so that starts to make us think then, are we actually seeing the reason why we're not getting those responses in certain areas of this tissue to that targeted drug is because effectively the cells are behaving differently because the context they're in is different. So therefore the signaling is different, right? And so we took a little fragment of this and we thought, wouldn't it be cool if we could embed agents as cells in that little fragment and take our 500 virtual cells and then just simulate what they would do if they were close to stroma, if they were far away, how would they behave? And we came up with this little sort of cartoon, overly simplified network. And so essentially we would seed that background sort of non-tumor area with random cells from our 500, right? And then basically, if they were close to the HGF, close to the stroma, then they would get the stromal network, under that, versus if they were far away, they would get the different network. And so basically what I'm going to show you here, and it's kind of tricky to see, but this is what happens when we look at the individual parts of that pathway. And what you can see in particular, if you look at the MET, right, that was one of the things that seemed to jump out in those stromally stimulated cells. There's yellow all around on the edge because that's right beside the stroma, right? Whereas it's blue where it's not. And then you've got kind of some random mixes there. We just let it do what it wanted to do. Now, if I run it on in time, we're getting to see how there's a whole bunch of that network that isn't doing hellish much, or at least they all seem to behave in the way. But there's quite a lot of dynamics happening in the MET, and the EGFR's got quite a distinct area. And one of the things you see is the EGFR is much higher away from stroma, and the MET's much higher right beside the stroma. So if I was going to throw on an EGFR inhibitor, potentially I'd get one part of that, but I wouldn't get the other, right? So at least that was the implication. So after a lot of wrangling, we managed to get this published in PLOS CB, and we're very in PLOS Biology, so we're very happy about that. But I'm intrigued about with the newer technologies that we have now, and things like spatial transcriptomics, and how one should maybe interpret that data. If do we just treat it as we're going to ignore the fact that you're right beside that stroma or that blood vessel, or do we actually have to filter out the fact that you're beside that blood vessel or that fibroblast, because it's going to change your signaling? Okay, so I've managed to get through that in quite good timing, I think. Did I? Yeah. So Unjung was the one who did all the signaling work. Thanks to Eric for allowing us to do that in his lab. David played a key role along with Simon Hare and Gustavo for the prostate cancer work. And so to conclude, tumor stroma dialogue modulates heterogeneity in many ways, including signaling, context, and potentially, even making you grow better or evolve quicker or slower. Stromal independent tumors seem to evolve faster, at least in our simulations. And there's a type of prostate cancer, and I can't remember what they call it, but we called it stromal independent. That's the most aggressive form of prostate cancer, and the origin of it's not clear. But what is clear is that there's no stromal cell at all in the prostate. They're all gone. They're just not there. And so you're like, how's that even possible, right? And so we actually think that if you've got a very poor stromal environment for whatever reason, could that drive this very aggressive phenotype that then effectively just pushes out that stromal population and just takes over? It's just a carpet of cancer. Stromogenic tumors grow faster but evolve slower. So the irony here, and this was again what our pathologist colleagues didn't want to hear on some level, but once he saw how it actually explained his data, he was kind of fine with it. But that reactive stroma could actually be a good thing on some level. Even if you can't treat it anymore, it might not be as nasty as you think it was going to be. We think that reactive stromal grade should become part of Gleason's scoring system. It should be there. And we already know a few pathologists who use it, and partly because of the work that Gustavo did independent of us. And then I wanted to come back to this idea of looking at populations or distributions rather than individuals. And with the kind of technologies that we have now of going down to single cells and dragging those cells out of context and then lining them all up and stacking them according to what they might be expressing, there's a danger with that, which is that you're actually finding patterns that might not really be there, right? Because you're ignoring where they're actually clustered. Now, maybe there's still a big enough signal that it's going to be useful, but I think we just have to be careful about that. And that idea then, okay, so there is a whole distribution of signaling that's going on. It's not just binary, right? And so we have to look at that, those drugs that target, especially targeted drugs for pathways, what they might be doing when you've got a highly expressing cell versus one that barely expresses it. And, you know, just to really emphasize what I'm trying to pitch here to you, hopefully, is that we need to understand the normal and the normal tissue architecture better. You know, we had a bit of a discussion about stem cells, et cetera, right? And in many ways, those are critical players in the normal architecture of many organs. But there's still some debate over some of these aspects. And I'm not just talking about stemness, but, you know, the whole normal form and function probably before the genome revolution was almost as important as, you know, understanding what the tumor was doing, right? So back in the days, development and sort of cancer were much more hand in hand, and genomics kind of pushed that apart. I think it's starting to come back because, of course, that data is pointing towards that, right? It's pointing towards de-differentiation and redifferentiation. And so really this idea of homeostasis being a key regulator, constraining the system, and then the cells have to do something special to break out of it, and how interactions and feedback with that normal homeostatic structure will potentially change the outcomes in significant ways. So I just want to finalize by thanking the NCI for funding us through 3 decades of PSOC and CSBC. The math onco community wouldn't exist without you. So we're greatly appreciative of that. And this is the review that I spoke about. We managed to even make the cover of Trends in Cell Biology thanks to Jeff's wizard skills. So we were pretty chuffed about that. You can read ABMs in integrative science, which would probably be pertinent to what this talk would be about. And that prostate paper above and the one in the middle is the context signaling. And thanks everybody for listening.
So before anybody else has a question, I'm going to jump in. Hi. I was intrigued this time watching this version of the talk. I've seen bits of pieces of this before, but pulling this together really made me think about connecting to some of the questions I was talking about during the didactic about the understanding if there are, let's say, 6 differences in the pathway, picking on pathways, or, you know, in these tools, but the pathways could be relevant. So it struck me that with HAL being around now available, and you can make lots of models very quickly, could you, some sort of bonanza effusive amount of models somehow generate to you the possibility to be able to back calculate out the potential differences in those networks?
Yeah, possibly. I mean, I think what you're saying is
with fairly limited data.
Yeah, with like hundreds of thousands of possible networks, but you've got the specific biopsy and of what those mixes gave you in your signaling or something, or would it be single cell, or would be bulk?
I'm thinking bulk, but I was just thinking like sex differences there's lots of data that's out there on this side. Could you use limited tissue data to back calculate how the pathway models
if we could create that network based on like what we know should be changing, for example, right? Then that would be a possibility, I think,
Hey, Sandy.
Hi there.
Great talk, as always. It was really interesting for me when you had this like minimal network, right? And you were like, well, there's actually multiple different parameter fits and multiple different regimes that we can all get the same result. They all fit equally well. Typically, my thought would then be like, oh, that's frustrating. But you were like, that's actually a strength. Let's turn that into like, now that's our kind of like almost like a Bayesian approach to these parameters. And like, I was just kind of curious if you could speak a bit more to that. Like, is that something you've seen elsewhere? I've never seen that before.
Well, you know, it's something we actually use when we're trying to fit models to patient data, right? Mm-hmm. So we create these virtual cohorts of fits because there's uncertainty in the parameterization. You can't say for sure that higher birth rate and that lower death rate or the inverse of that is equally fitting your data. Which one of those is it? So if you use that cohort instead, that ensemble, that, you know, distribution, and now you see that when you apply your treatment or whatever you're trying to predict, that it segregates into two very clear behaviors, and the ones that didn't perform so well had this mix of parameters or signaling, and the ones that did perform well had this. And so that then gives you some insights to specifically look for that in the data. Yeah. So like we've used it as a means to try and calibrate our models and to constrain. So if you now imagine that you can predict forward not only this time point, but the next one, and on that next time point, you've got an imaging piece or an extra biopsy, whatever it would be. So all that uncertainty now narrows a bit more because you threw out all this stuff that didn't match, right? Yeah, so that's one way to think about it.
Thanks.
Sandy, excellent work.
Thanks.
I was just thinking by looking at our paper in Nature Equilibrium, we know that younger persons don't get cancer as often, and when they do, it's actually a lot more aggressive. Is that consistent with results of that model? Is that the way you're thinking about this as well?
Well, you know, I didn't show. We've got this melanoma story that looks at senescent fibroblasts, right? So, the models that I showed say nothing about the stroma except that it'll just become reactive because some secreted factor. But we actually know that fibroblasts, as we age, can become this, what do they call it, a secretory phenotype, which is essentially like a reactive type of phenotype. And so that would potentially facilitate then that transition.
But that would make it very, which is great. Is the mechanism by which this happens. But basically, you look at the missile model. The idea is that you have permissive environment, non-permissive environment. It's very abstract. It's about prostate cancer, but actually can model lots of different things. You have a permissive environment. A tumor doesn't really need to be very aggressive to use the things. Older people maybe have more permissive environments.
Yeah.
Younger people have probably tougher environments.
Yeah, that's true.
For a tumor to come out,
it's going to be worse.
Yeah. So is this something consistent with that?
Yeah, I mean, that would be consistent with that. It's a grim reality that is the fact that we have looser controls better for our cancers than, you know, our more restrictive younger cells, which if you get cancer there, it's going to be damn nasty, basically. That's the implication, right?
Yeah.
Yeah.